You're welcome to this session at NaftaliMuhumuza.com. And in this session, we want to talk about renal tubules. In the previous video, we looked at renal corpuscle, and we saw glomerular filtration, and we also looked at the regulation of GFR. So in this session, we want to talk about renal tubules. And in renal tubules, we see majorly reabsorption and the secretion. But before you begin, always make sure you subscribe if you haven't subscribed. Like the video after watching and share your comments. So in renal tubule, we are seeing renal tubules, and these renal tubules we shall see is composed of the proximal convoluted tubule, then the loop of Henle, which is divided into the descending limb and ascending limb. Then we also have distal convoluted tubule. Then lastly, the distal leads into the collecting duct. So these are the parts of the renal tubule. We saw in a recent video the parts of the renal corpuscle was glomerulus and the Bowman's capsule. And we saw how filtration occurs across the glomerulus. So today we want to look at selective reabsorption. We formed the glomerular filtrate. Then we want to see how substances within the glomerular filtrate are selectively reabsorbed. So after filtration, we shall find glomerular filtrate was composed of electrolytes. We're having there glucose. We're having amino acids. We're seeing there glucose, then sodium they belong here, whereby we have sodium, potassium, hydrogen. These are electrolytes. Bicarbonate. We have the phosphate, the hydrogen phosphate, the chloride, all are under electrolytes. We saw substances like creatinine, waste products of metabolism like creatinine, urea, uric acid being part, even para-aminohydric acid, all were part of the glomerular filtrate. That after filtration, these are, that because they are smaller in size, we find them, they were filtered via the penetrations and the filtration is late. Then we saw plasma proteins and blood cells not being reabsorbed. So we don't expect them to find them in the glomerulus. So we want to see how these substances, as they pass through proximal, as they pass through loop of Henle and the collecting duct, how are they reabsorbed, depending on the body's need. And we want to begin with proximal. We want to see reabsorption under proximal convoluted tubule. And in the proximal, we see this proximal convoluted tubule part, which is near the Bowman's capsule. It has numerous brush borders. And then it contains the mitochondria that provides energy in the form of ATP. That during active transport, we see them playing a good role. And this transport or reabsorption of these substances in the PCT, they occur by three processes. We shall see processes occurring whereby we shall have passive diffusion. They can be reabsorbed by passive diffusion. They can be reabsorbed by active. And we shall see this active transport shall be majorly secondary. Secondary active transport where the ATP is indirectly involved. Then the passive they can pass via either those ones that cross the cells. We call them this route, we are going to call it transcellular route. Transcellular route. And there are those that pass through the tight junction, and this route we are going to call it paracellular route. So some substances they move across the cells to enter into blood circulation. Others use the gap junction to enter into blood circulation. So that is what you call transcellular and paracellular route. And then we have seen this proximal convoluted tubule having numerous brush borders that increase the surface area for reabsorption. That's why we are going to find out that in the proximal, that is where most of reabsorption occurs, because it is highly coiled into brush borders. Its columnar epithelia is full of brush borders, which increase the surface area for reabsorption. And another thing, it contains mitochondria, which provides energy in the form of ATP. And we're going to see substances that are absorbed here. Number one is sodium. We're going to see that most of the sodium, greater than 70%, is reabsorbed in the proximal convoluted tubule. And it is by active transport, whereby we see this sodium being re-exchanged. It moves into the tubular tissue, and as it is moving, it is exchanged with hydrogen. Then hydrogen moves. So as we excrete hydrogen from the tubular cell into the lumen, we see sodium being exchanged, and this one occurs in the presence of ATP. This one occurs in the presence of ATP, whereby ATP is hydrolyzed to ADP plus phosphate inorganic. So that's why we call it active transport. So we are seeing reabsorption of sodium. Sodium first is being reabsorbed by a transporter known as sodium stroke hydrogen antipode. We call it antipode because they are moving in opposite direction. We are seeing as sodium is being reabsorbed, it is exchanging with hydrogen. So we call it an active transport known as sodium stroke hydrogen antipode, which is a form of secondary active transport. That is how we are able to reabsorb. And we are seeing the majority of sodium being reabsorbed in the proximal. This is substance number one, which is reabsorbed. And as sodium is also moving, we are seeing also urea, not urea, but amino acids. Amino acids, they are also reabsorbed by active, that when sodium moves into the cell of the kidney tubules, it creates an osmotic gradient that also facilitates the movement of amino acids. So we're going to say that amino acids are reabsorbed by active transport first, in the presence of ATP secondary active transport, but later they move along the electrochemical gradient. So it is both active and passive, that is amino acids. Then we are going to see other substances like glucose. That when we have glucose, we see majority of the glucose also being reabsorbed in the PCT by active transport. That whenever we have sodium inside here, as the sodium is moving in the cell, the glucose says I will not remain behind. It jumps on it, and also they move together into the tubules. So we are seeing the mechanism of absorption of glucose is also a secondary active transport, whereby glucose is reabsorbed by a secondary active transport known as sodium stroke glucose co-transport, or you can call it a symporter. This one we call it antipode because they are moving in opposite direction. This one is a symporter or a co-transport because they are moving in the same direction. So we are going to see this is also an active transport. And most of the glucose is being reabsorbed by this. And sometimes we call this form of transporter, we call it sodium-dependent glucose transporter type 2. And we shall see many drugs which target it in diabetic patients. But when someone is diabetic, they can easily give them this drug which target sodium-dependent glucose transporter type 2. And as we move on, we are going to see that apart from these processes, we are going to see even how do we reabsorb, how is bicarbonate reabsorbed. If this is my proximal convoluted tubule, we are going to see that bicarbonate ions, we saw sodium exchanging with hydrogen. So when sodium exchanges with hydrogen, as sodium is moving, hydrogen is coming in from into the cell, into the lumen, and this hydrogen comes and binds with bicarbonate to form weak carbonic acid. This is in the presence of carbonic anhydrase enzyme, and this weak carbonic acid dissociates to give water plus carbon dioxide. So we are going to see that when hydrogen in the lumen combines with bicarbonate in the presence of carbonic anhydrase to form weak carbonic acid, which can give out water and carbon dioxide. And we are going to see that this water and carbon dioxide enters into the lumen, enters into the tubular tissue, whereby water combines with carbon dioxide to form again weak carbonic acid. And this weak carbonic acid dissociates to give hydrogen ions plus bicarbonate. And these bicarbonate are absorbed in the blood and they are exchanged with chloride. Since we lose the negative charge from inside into blood, we see chloride shift coming in to neutralize the charge. Then the hydrogens which are produced, they go back into the lumen. So in summary, bicarbonate ions in the lumen are reabsorbed as carbon dioxide, in the form of carbon dioxide. And after being reabsorbed in the form of carbon dioxide, when they reach in the tissue, they combine with water to form weak carbonic acid, which dissociates again to give bicarbonate plus the hydrogen ions. Hydrogen ions coming back, then the bicarbonate enters blood. So that is the way how we absorb the bicarbonate. We absorb them in the form of carbon dioxide, facilitated by an enzyme known as carbonic anhydrase. So this is what is happening in the proximal convoluted tubule. Lastly, under reabsorption in the proximal convoluted tubule is water, whereby we see even water is reabsorbed. Water is also reabsorbed by passively or by what we call obligatory diffusion. It is a form of osmosis. Obligatory means it is not being helped by any. It is just moving along the concentration gradient or being attracted by the osmolarity. That is what we call obligatory. But facultative form is whereby antidiuretic hormone is involved to enable reabsorption of water. But in the proximal, we see obligatory reabsorption of water, whereby it is being reabsorbed against the concentration or along the concentration gradient, depending on the osmolarity of the blood. So this is what we can discuss in under reabsorption in the proximal convoluted tubule. So from here, we can move into the loop of Henle. Loop of Henle, which is a hairpin loop made up of the descending limb and ascending limb. The descending limb is thin, the ascending limb has a thick part, what we call the thick ascending. And we shall see majority of the reabsorption being happening of solutes in the thick ascending. That in the loop of Henle, the descending limb of loop of Henle is permeable only to water. It is permeable only to water due to the presence of aquaporin type 1. The aquaporins are the one that allow reabsorption of water from the tubules into the interstitium. So we see reabsorption of water into the interstitial space in the descending limb of rope of Henle. So if I have rope of Henle here, this rope of Henle, we are seeing that in the descending limb, it is reabsorption of water. Water is reabsorption because this descending limb is mostly permeable to water. It's permeable to water due to presence of aquaporins. These aquaporins are the one that enable reabsorption. So we see it in the descending limb, there is no reabsorption of water. There is no reabsorption of solids, only reabsorption of water. Why? Because the descending limb is permeable only to water but impermeable to electrolytes. So only reabsorption of water is seen in the descending limb of rope of Henle. And then when we go to the ascending limb, especially the thick ascending limb, you're going to see a transporter that if we have this as our ascending limb, we see it has a transporter which takes in sodium, and as sodiums are entering inside, sodium moves together with potassium, and when also chloride sees potassium has entered, we see also reabsorption of chloride, and there are two of them. So chloride also enters together with them, two chlorides enter together with them. And this form of transport is a secondary active transport, and this secondary active transport is known as sodium stroke 2 chloride stroke potassium co-transport. That is a secondary active transport. That in the thick ascending limb of rope of Henle, we are seeing as sodium is being reabsorbed via transcellular route. Two chlorides move with it and one potassium. And when potassium accumulates this side, we see it coming back via the paracellular route. So if there is another cell here, we see potassium coming back into the lumen via paracellular route using paracellular. When it was going to use the transcellular route, then when it is coming back, it uses the tight junction to come back inside the lumen of the thick ascending. So the form of secondary transport in thick ascending limb is sodium stroke 2 chloride stroke potassium co-transport, which is a form of secondary active transport. And that's why we are saying we have seen in the descending limb permeable to water, impermeable to solids. Then we see the ascending limb permeable to electrolytes or solids, but impermeable to water. We don't see reabsorption of water within the ascending limb, especially thick ascending limb. We see only reabsorption of electrolytes, but not reabsorption of water. Then in the descending, reabsorption of water, no reabsorption of electrolytes. So this, when we come back here we are losing water into the interstitial space and back into blood. Here we are not losing water, but we are losing only sodium, potassium, chloride. Those are the electrolytes. And this countercurrent flow, that as you're moving down, you're losing water. As you're moving up, you are not losing water. You are only losing solids. It creates the osmolarity, or it creates what we call the osmotic gradient within the interstitium, which is responsible for concentration of urine, as we shall see it in the next video. This is what we call countercurrent multiplier effect of the rope of Henle. That as the solutes are moving here, they are being concentrated, where it starts at 3,300 being isotonic, it increases to 600. By the time it is at the hairpin rope, it's at 12,000, and as it's going back, still it reduces due to the countercurrent multiplier effect of the loop of Henry. And this osmotic gradient created by the countercurrent mechanism or countercurrent multiplier of the loop of Henry is maintained by the countercurrent exchanger, by the vasorector, which we shall see in the next video. How the loop of Henry enables us to concentrate urine, to conserve water during dry condition and strenuous exercise. That is how we shall look at in the next video. As we conclude this, the last part of reabsorption is the descending limb, whereby descending limb of rope of, no, the last part is the distal convoluted tube. And in the distal convoluted tube, it is divided into two. The first part and the second and the late. There is early part of the DCT and the late part of the distal convoluted tube. And major reabsorption, we see it in the late part of the distal convoluted tube, whereby in the late part of the DCT, we find cells like principal cells. It has principal cells, it has intercalated cells. And we see these principal cells being the one, we know that naturally DCT is impermeable to water. It is impermeable, the red part is impermeable to water. But whenever there is need of water, maybe there is little intake and there is much exercise you have been doing, you have been sweating, the body needs to conserve water. The increase in osmolarity, it triggers the osmoreceptors. It has the osmoreceptors, and these osmoreceptors detect increase in osmolarity here and sends impulse to posterior pituitary gland. And this posterior pituitary gland is the one that releases ADH, or what we call vasopressin. And when ADH is released, it is the one that comes and binds on the receptors of the principal cell and causes them to reabsorb more water. So more water is reabsorbed in the late part of the distal convoluted tube, whereby the antidiuretic hormone, or what we call the vasopressin, is the one that binds on the principal cells, leading to reabsorption of water, hence producing concentrated urine. That is when we see ADH coming in. And still in the late part, in case we need more sodium is needed, if more sodium is needed, we see it also being reabsorbed. That more, if there is little sodium in the blood, it triggers the adrenal cortex. And this adrenal cortex releases aldosterone hormone. And this aldosterone hormone is the one that causes the reabsorption of sodium. If more is needed in the body, we see more reabsorption. And still we see also, in case we need more calcium in the body, it is reabsorbed under the influence of parathyroid. That this parathyroid hormone, produced by the parathyroid gland, found in the neck region behind the thyroid, is also responsible for reabsorption of more calcium in the late distal convoluted tube. That is the reabsorption in the distal convoluted tube. And this same mechanism in the late part is the same mechanism in the collecting duct. We shall also see that in the collecting duct, there is more, if more water is needed, is reabsorbed. We shall see water being reabsorbed. We shall see that even if more urea is needed, is reabsorbed in the collecting duct. So in the collecting duct, we see reabsorption. We see reabsorption of water and urea. Water under influence of ADH. And we told you in the beginning, whenever ADH or antidiuretic hormone is involved in reabsorption, we call it facultative reabsorption of water. So water is being reabsorbed by facultative. This is what we call facultative reabsorption of water being under influence of ADH. So water is reabsorbed in the DCT and the collective duct by facultative means in the presence of antidiuretic hormone. Because we know naturally distal convoluted tubule and the collecting duct, they are impermeable to water. So they are only become permeable in the presence of ADH, what we are calling facultative reabsorption. So this is what we can talk about selective reabsorption. We have seen selective reabsorption under proximal. We have seen it in the descending limb, whereby we said in the descending limb it is only majorly water, because it is impermeable to solutes but permeable to water. Then we saw in the thick ascending of loop of Henry, being impermeable to water but permeable to solutes like sodium, like potassium, like chloride. And that was also a secondary active transfer. But we have seen in the distal convoluted tubule, in the left part, having principal cells, which are responsible for facultative reabsorption of water, whereby they stimulate production of antidiuretic, and antidiuretic hormone causes reabsorption of more. And we have seen even more, if more sodium is needed, is reabsorbed under the influence of aldosterone hormone, produced by the adrenal cortex. We have seen also, if our body needs more calcium, this calcium is also reabsorbed under the influence of parathyroid hormone. And we have seen lastly, in the collecting duct, also another facultative reabsorption of water under influence of antidiuretic hormone. Then urea passively reabsorbed. We see urea is passively reabsorbed in the loop of Henle and in the collecting duct. So the last part we can talk about here, is secretion, because we saw the renal tubules play a role in reabsorption and secretion. So the second part, the first part we have finished is renal reabsorption, selective reabsorption. Now we want to look at secretion, renal secretion. And renal secretion, we see the major substances which are secreted by the renal tubules. In the PCT, we see secretion of hydrogen in exchange for sodium. We see secretion of para-aminohydric acid. We see secretion of penicillin. If you have been on penicillin drug, we see also secretion of ammonia. And how is ammonia secreted? That in the tubules, if we have the tubules, we have glutamate. Glutamine is deaminated in the tubules. We see glutamine being deaminated to produce ammonia. And this ammonia diffuses into the lumen. And you remember in the lumen there is exchange of sodium, that when sodium enters in the cell, we see hydrogen ions being secreted. And this hydrogen ion combine with ammonia to form ammonium ions. And this is how the proximal is able to secrete ammonia in the proximal. So we are seeing in the proximal, we are seeing a reabsorption of ammonia, secretion of ammonia and secretion of hydrogen. Then plus also secretion of para-aminohydric acid and penicillins being secreted to be part of the lumen. So they become part of the lumen, or what we call the glomerular filtrate. Then in the loop of Henle, we see major secretion of urea. Because urea is both secreted and both reabsorbed. Then in the ascending part of limb, we we see secretion of potassium. We saw here that potassium is reabsorbed, but when it reaches in this side, it comes via paracellular route. And we see it being reabsorbed. That is potassium. Then the DCT, majorly we see also secretion of hydrogen, secretion of drugs like penicillin continue to be secreted. And that is what we can talk about under renal secretion. So substances which are secreted by the kidney is hydrogen ions, para-aminobutyric acid, penicillins, ammonium ions, urea, potassium are the only substances that are secreted to be part of the tubules. And as we conclude, there is also another form we can talk about in here. That these substances which were being reabsorbed, they are categorized into three, whereby there is what we call high renal threshold substances. We have what we call low renal threshold substances. And we have non-threshold. Non-threshold substances, these ones are they are not reabsorbed at all. These ones are not reabsorbed at all. The way they enter into the glomerular filtrate, the way we excrete them in urine. Those are non-threshold, and example here is creatinine. Creatinine is neither reabsorbed nor secreted. The low renal threshold substances include amino acid, include uric acid, include urea. These ones, they can even be found in urine in normal condition. These ones, the one with low threshold, they are found even in urine in normal conditions. Those are low threshold, like uric acid, like urea. So that even for them, even under normal conditions, they are found in urine, but they are still reabsorbed. But they are also excreted and they are found in urine in normal conditions. Those are what we call low threshold. Then high renal threshold, these are the ones that are not found in urine under normal conditions. Under normal circumstances, these substances are not supposed to be found in urine. They are only found in urine when they reach renal threshold. For substances like glucose, it is not supposed to be found in urine, but it can only be found in urine when the blood concentration goes beyond the renal threshold, whereby its renal threshold is 180 milligrams per deciliter. That we can only find these high renal threshold substances when they go beyond the renal threshold. That's when they can be found in urine. So they are found in the urine under abnormal circumstances. That's what we call high renal threshold substances. And we have seen the renal threshold of glucose is 180 milligrams per deciliter. So in summary, high renal threshold, in normal conditions, they are not supposed to be found in urine. Only found in urine when they are exceeding their renal threshold. Then the ones of low renal, for them, they are naturally found in urine, even under normal circumstances. They are both secreted and both reabsorbed. Then non-renal threshold, these ones, they are not found. They are not reabsorbed at all. So they are all found in the urine because they don't undergo. The way they enter in the glomerulus, the way they are excreted. And an example is creatinine. Thank you so much for listening. Always remember to share the video. Always remember to subscribe if you haven't subscribed. We are always at your service. Thank you so much. In the next video, we shall look at how to produce dilute and concentrated urine by the kidney. Thank you so much.